Right, first question then is an AO1 question. It's a recall knowledge question asking us to look for two differences or state two differences between mitosis and meiosis. So if we look at the mark scheme, we can see that mitosis has a single division where one parent cell forms two genetically identical daughter cells. Now in meiosis, just underline that, genetically identical, that's mitosis. In meiosis, there's two separate divisions, meiosis 1 and meiosis 2. And we know 4 cells are produced from meiosis versus only 2 daughter cells from mitosis. We know the gametes produced from meiosis are genetically unique. And we have to have the word genetically in that answer for mark point number 2. Right, with mitosis, we know because the daughter cells are genetically identical, if the parent cell is diploid, the 2 daughter cells will also be diploid. But also with mitosis, if the parent cell happens to be haploid, then the 2 daughter cells, because they're genetically the same, will also be haploid. Now with meiosis, when we talk about the chromosome number, if the parent cell is diploid, that means 2 sets of chromosomes. The daughter cells, which are often gametes, will all be haploid. So we know meiosis halves the chromosome number. For example, if you take a germline cell that lines the testes or the wall of the ovary in humans, that's got 46 chromosomes. The gametes will have 23 chromosomes. So 46 to 23 is half the chromosome number, and it's gone from diploid to haploid. Now the gametes, as we know, will be genetically unique. So if you look at mark point number 4, it says in mitosis, the parent cell is diploid and the daughter cells are diploid, or if the parent cell is haploid, the daughter cells will be haploid. In meiosis, the parent cell is diploid and the spores or the gametes will always be haploid because it halves the chromosome number. Now in meiosis, there's separation of homologous chromosomes. So you'll have a bivalent that forms in prophase 1, and the 2 homologous chromosomes each are made of 2 identical sister chromatids. The intact replicated homologous chromosome separates to opposite poles of the cell, but that does not take place in mitosis, it's only in meiosis. So there they needed the separation of homologous chromosomes from the bivalent. Right, also in prophase 1 of meiosis, we have crossing over. Now, crossing over is where an equal length of non-sister chromatids is swapped over, and that occurs at the chiasmata point. So I just put chiasmata point. Now we know that produces new combinations of alleles from crossing over. Crossing over does not take place in mitosis. So this only occurs in meiosis and not in mitosis. And this will produce a new combination of alleles. We also know that the bivalents, because they attach to separate spindle fibers independently of each other, when the homologous chromosomes separate to opposite poles of the cell or segregate to opposite poles of the cell, that's a random process. So independent segregation occurs in anaphase or metaphase anaphase 1 of meiosis. That does not occur in mitosis. So in mitosis, there's no crossing over and there's no independent segregation of homologous chromosomes. Okay, in this question, we're going to look at meiosis then, and it tells us that at the end of meiosis, so we are going to do a bit of annotation, that these fungal cells, these diploid fungal cells, produce spores which are haploid. Now we know the spores must be haploid because they've been made by meiosis. So from every parent cell that's diploid, diploid means 2 sets of chromosomes, we get 4 spores which are haploid with one set of chromosomes. Then it says the spores are collected in narrow tubes, and that restricts their movement, so we can look at them and count them. And the spores can either be a pink colour or a white colour. So the colour of the spore is determined by a single gene, which is located on a pair of homologous chromosomes. So the locus is the position of the gene. Now there's two variants of this gene, and a variant of a gene is called an allele. So there's one allele for the pink colour, and there's another allele for the white colour of the spores. Now, if you look at the zygote in figure 7, it does tell us that there's a pair of homologous chromosomes. So if you look at the chromosome on the left, I've just labelled that number 1, and then you look at the matching chromosome on the right, so I've labelled that number 2, we can see that these chromosomes are the same size, they have the same gene at the same position or locus, therefore they must be homologous. So that zygote is diploid, it's got two sets of chromosomes, and this diagram is only showing one type of chromosome, of which there are two versions. Now, if you look at chromosome 1 on the left, hopefully you can see that this chromosome is made of two identical sister chromatids. So this is a replicated chromosome. It's still counted as one chromosome, but it's after DNA replication, which occurs in S phase. So you can see that hopefully there's two identical sister chromatids, which are two separate but identical molecules of DNA attached to the centromere. So if you look at the allele on the left here at that locus, because the other sister chromatid is an exact duplicate of the first molecule of DNA, there's going to be that identical pink allele on the other sister chromatid. So this is one replicated chromosome, and we can see it because this is obviously after meiosis. Now the matching or the homologous chromosome, instead of the pink allele, we've got the white allele. Now because there's two identical molecules of DNA, on the second sister chromatid there's also the white allele. Now these two homologous chromosomes would pair up during prophase 1 and they would form a bivalent. Now we know during the first nuclear division, that means meiosis 1, that the homologous chromosomes separate to opposite poles of the cell, and we then get cytokinesis. So we can see that chromosome number 1 has separated into the left-hand daughter cell, and the intact chromosome number 2 has separated into the other daughter cell. So in meiosis 1, we know the centromere does not split. The sister chromatids are not pulled to an opposite pole to the cell in a V shape, but the intact replicated chromosomes move to opposite poles of the cell. Now, the second nuclear division would be, for example, where the sister chromatids are pulled to opposite poles of the cell. So if you look at this diagram here, we've got this replicated chromosome. One sister chromatid would go to the left of the cell. The other sister chromatid would be pulled in a V shape to the opposite pole of the cell. So this spore here, if you look at it, would have one sister chromatid with the P allele or the pink allele. The second spore would have the matching other identical sister chromatid, again obviously with the pink allele. This replicated chromosome that I've just circled in on the right hand side, the two sister chromatids would separate, they'd be pulled to opposite poles of the cell, the centromere would be split, and therefore we would get one sister chromatid in that spore with the white allele and the identical sister chromatid in the fourth spore, again with the white allele. So the allele determines the colour of the spore. So we can see there's a single sister chromatid in each spore. That can now be called a chromosome again. So they are haploid. We could say haploid means one set of chromosomes, and the allele determines the colour. So obviously we could use the key there because it says this one here is a pink spore and the other one's the white spore. So hopefully you can see that meiosis 2 down at the bottom is very similar, very similar to mitosis. Now we know that as the spindle fibers contract, the centromere splits and the identical sister chromatids are pulled to opposite poles of the cell in a V shape. So these cells here at the end of meiosis 1, they are actually both haploid cells, but the one copy of that chromosome is still replicated. So the cells at the end of meiosis 1 are haploid, and the cells at the end of meiosis 2 are also haploid, but they'd have half the DNA mass because each chromosome is not made of two sister chromatids. It's only a single chromatid in the spores at the bottom. So the first question says there's 7 chromosomes in a spore nucleus. So we know the spore nucleus is haploid. And it says place a tick in the box next to the number that represents the number of chromatids present in the zygote shown in figure 7. So it's talking about the number of chromatids. So that's really, really important on the left. Now, if you look at the zygote in figure 7, we know it's diploid. That means there's going to be 14 chromosomes because 2 sets is 14 and 1 set is 7. So 1 set would be 7 at the top. Now, if you have 14 chromosomes instead of 2 chromosomes that we saw in the zygote, we know from figure 7 that each chromosome is replicated and made of 2 sister chromatids. So if we were to draw out all 14 chromosomes, okay, each one would look like that because each chromosome is replicated. So hopefully you guys can see that if there's 14 of these, there's going to be 28 chromatids. So that was the answer for 1 mark. So 7 chromosomes is haploid, the zygote is diploid, that's 14 chromosomes, but using the diagram that we're given in figure 7, we know that each chromosome is replicated. It's made of 2 identical sister chromatids. Each sister chromatid is just 1 molecule of DNA. So we get 2 molecules of DNA joined at the centromere because that's after DNA replication. So we're going to have 28 chromatids in our zygote. Scientists then recorded the arrangement of coloured spores inside many narrow tubes. So we're going to use the data here and the headers. Now, hopefully you guys can see that the number of narrow tubes with the first arrangement, that's pink, pink, white, white, is 81. And if we look at the opposite arrangement of white spore, pink spore, that is 78. So these two numbers are actually very similar. Now you're going to get the pink, and the white from what we just saw in the previous diagram. But we're going to get white, pink, pink if the bivalent, so imagine this was the bivalent, the first one. Can you see that the pink alleles are on the left and the white alleles are on the right? That would be the bivalents that would give rise to this arrangement. Now, if that bivalent was arranged in the opposite way, with the chromosome on the right, on the left this time, what we'd have is this arrangement of the bivalent on the spindle. So the first chromosome made of those 2 identical molecules of DNA would be on the left, and the second matching chromosome would be on the right. So this arrangement here would give rise to the arrangement in tube number 2. You'd have the first spore on the very left with a white allele, the second spore would have a white allele, the third spore would have a pink allele, and the fourth spore would have another pink allele. Now we know this is called independent segregation and it's random because that bivalent can attach to the spindle fibre in either this arrangement number 1 or this arrangement number 2. So there's going to be a random shuffling of homologous chromosomes to either pole of the cell, and it depends upon which orientation the bivalent attaches to the spindle. Now, if you look at tube number 3, hopefully you can see that that value of 10, so we got 10 tubes with this arrangement of pink, white, pink, white. We can see 10 is actually a lot less than 81 and 78. Now, this is going to be due to crossing over. Now, we know crossing over is actually quite a rare event. It occurs in about 10% of cells that undergo meiosis. So if we draw out the same bivalent with the sister chromatids, if you had this to start with, so it was pink, pink, white, white, imagine there was crossing over. So what would happen with crossing over is this. You'd have pink, white, right. So hopefully you guys can see that there's been crossing over between an equal length of non-sister chromatid. So where the pink allele was, there's now a white allele, and where the white allele was, there's now a pink allele. So this is actually quite rare. That's why we get a fewer number of tubes with that arrangement of pink, white, pink, white. So it says, using all the information in the question, what can you conclude from the scientist's results about the movement of chromosomes in meiosis in this fungus? So if we look at the answers then, there's separation of homologous chromosomes, and that's called independent segregation, and that occurs in metaphase anaphase 1. The arrangement or the separation of homologous chromosomes is random. That's why you get an equal number of the spores in tubes 1 and 2 with their respective arrangements. Mark point number 3. So we've got to use the data. Crossing over has occurred in tube number 3. So we've got to reference tube number 3. And there's only 10 tubes with that arrangement. And that's because crossing over is rare or very infrequent, which gives the lower number of 10 versus 80 or 78 that we had earlier. So we've got two main sources of genetic variability in meiosis. We've got independent segregation of homologous chromosomes from the bivalent, and that is random. And we've got crossing over, where we get the chiasmata point and an equal length of non-sister chromatids swapping over to make new allele combinations, but that is actually quite rare. So we get a fewer number of tubes with that arrangement of pink, white, pink, white. Okay, in question number 8, we're going to switch to gas exchange in fish. So we're going to be talking about the gills. Now, figure 4 just shows us two main structures of the gills. So F would be the gill filaments, and G would be the gill plates or the gill lamellae that extend out from the gill filaments. So it wasn't accepted if you put gill arch. That was rejected. This structure down at the bottom would actually be the gill arch from which the gill filaments extend. So these are gill filaments, and then these plate-like structures with a big surface area are the gill lamellae or the gill plates. There you go, gill lamellae. Now, describe and explain the advantage of the countercurrent principle in gas exchange in fish. So there's going to be 1 mark for describing what happens, the countercurrent principle, and then explain the advantage of this system for these fish. So the first mark point then is the description bit that we can see there. We know water and blood flow in opposite directions. So water flows over the gill lamellae, and the blood flows inside the capillaries inside the gill lamellae in the opposite direction. Second mark point then is explain the advantage of this to the fish. So if you get the countercurrent mechanism with water flowing over the gills in one direction and the blood flowing in the opposite direction in the capillaries inside the gill lamellae, it's going to maintain a diffusion gradient of oxygen. So oxygen diffuses from the water into the blood, and that concentration gradient, although it is a small concentration gradient, it is maintained over the entire length of the gill lamellae. So if we do a little picture here at the top, imagine this was the water, and then we had the blood here underneath inside the capillaries. If the water was at 100 and then it decreased towards, say, 0, if the water is going from left to right and the blood is going from right to left, we can see that as the blood enters, there's no oxygen in the blood. So there's a small concentration gradient, but obviously the blood's going to pick up some oxygen, that goes up. However, if you look at the 20 value versus the 10 value, there's still a higher concentration of dissolved oxygen in the water. As the blood is going from right to left, it picks up more oxygen, but there's still corresponding higher dissolved oxygen concentration in the water. The blood keeps picking up its oxygen, it gets to 40, but we can see hopefully that the water content of the oxygen is still higher. So this concentration gradient, although it is a small gradient, is maintained across that entire surface of the gill lamellae. So the blood will get to around about 90% saturated. It's not fully saturated, but that's enough to meet the demands of the fish. So maintains a diffusion or concentration gradient of oxygen across the entire length of the gill lamellae. Okay, equilibrium is never reached, as we can see from the diagram at the top. If it was parallel flow, what you'd end up with is 50% O2 in the water and 50% O2 in the blood. That would be about halfway across the gill lamellae, so therefore equilibrium would be reached and diffusion would stop. So that would be the parallel flow system. But the question is obviously asking about the countercurrent principle. Okay, so if you look at some of the data on the graph then, we can see that the x-axis shows the water depth and the y-axis is the relative oxygen concentration. So you might be able to see that as the water depth increases from 0 to around about 750, there's actually a decrease in the relative oxygen concentration in the water. So the oxygen is dissolved in the water. Now, at a depth further down than 750 meters, all the way up to 1400 meters, we can see there's a slight increase in the concentration of dissolved oxygen. So 750 meters is the depth where there's the lowest concentration or relative concentration of dissolved oxygen. Now, if we look at the data in the table, we're given the 3 different species, A. fimbriae at the top, And it says the depth of water where this species of fish was caught is between 450 to 1280. So if you take your pencil and draw on some lines, 450 is the red line on the left, and 1280 is obviously the red line on the right. So hopefully you can see the distribution of A. fimbriae. It's always a relatively low oxygen concentration, but at 750 meters down, it can survive at extremely low oxygen concentration. Now, if you look at the second species, so A. fimbriae was species number 1, and C. melanoris is species number 2. So again, we're going to look at the data. The lowest depth is 850 meters, and the deepest depth is 1307 meters. So you only find species 2 between the two blue lines. And then we could also look at species 3. So species 3 is found at the deepest depths, 1205 to 1307. Now it's interesting to look at the ratio of gill surface area to body mass. So hopefully you guys can see that 148 to 1 is the highest ratio. So A. fimbriae has the biggest surface area of its gills in relation to its body mass. Now you might be thinking, obviously if it's got bigger gills, it's going to have more gas exchange, so the dissolved oxygen diffuses across the gills into the blood, and the CO2 from respiration diffuses the opposite way. And that's because obviously it needs to get more oxygen from the water into its blood when it's located at 750 meters, which is the lowest concentration of oxygen in the water. So it says use the information from figure 5 and table 4 to describe the relationship between the dissolved oxygen concentration and increasing water depth. So you really had to look at the 750 value being the depth with the lowest O2 concentration. So if we just quickly look at the answers then to number 1. Okay, so 750 was the key there. You had to reference that. They did accept 730 to 790, sorry. Okay, so the oxygen concentration decreases down to 750 meters, then further down than that, the O2 concentration will increase again, so it will go up. Now, number 2, dissolved oxygen concentration and the distribution of A. fimbriae. That was the first species in the table. So A. fimbriae is actually present in quite a wide range of different oxygen concentrations. It's fairly low oxygen concentration, but it goes down to that very low value at 750 meters. Or you could have said A. fimbriae is present in water at the lowest oxygen concentration at that 750 meters. Okay. So the other two species are never found at 750 meters depth because the oxygen concentration is too low at that depth for the other two species, because their gill surface area to mass ratio is not big enough. Okay, question 8.4 then, it says using information from figure 5 and table 4. So again, we're using the graph and the table, we're using the headers and the data. What can you conclude about the adaptation of the gas exchange surfaces of these 3 species of fish? So I would definitely start with A. fimbriae and say the lower the oxygen concentration in the water, the O2 is dissolved in the water, the higher the ratio of the gill surface area to body mass. And this is really important because it is asking us to what conclusions can we make about the adaptations of the gas exchange surface. Obviously, if there's a bigger surface area of the gills, that allows more oxygen to diffuse across them from the water into the blood. And that's really important because it supplies enough oxygen to maintain the respiration rate of that fish. So it's trying to link the oxygen is needed as the final electron acceptor inside the mitochondria of the cells in aerobic respiration. Okay, 9.1, describe and explain the effect of increasing carbon dioxide concentration on the dissociation of oxyhemoglobin. So this is what we call the Bohr shift. So imagine you have the typical sigmoidal shape graph. On the x-axis, you've got the partial pressure of oxygen. That just means the concentration of oxygen in a mixture of different gases. And on the left, you've got the percentage saturation of hemoglobin with oxygen. So we can see at low ppO2, this is in the absence of carbon dioxide, there's going to be very few hemoglobin proteins with oxygen bound to them, and most is actually being released. So there's a very low percent saturation. At high ppO2, at the right-hand side of the x-axis, there's a very high percentage of hemoglobin with oxygen bound. So most of the hemoglobin will be as oxyhemoglobin, which is this point here over the right. Now, if you add carbon dioxide into the mixture, and CO2 is a waste gas from aerobic respiration, that sigmoidal shape curve shifts to the right-hand side. So what happens is the carbon dioxide lowers the affinity of hemoglobin for its oxygen, and therefore there's more dissociation of oxygen from the oxyhemoglobin in the presence of carbon dioxide. And so the percentage hemoglobin saturated with oxygen is going to be lower because a lot of the oxygen has been dissociated and released, where it's then going to be used in aerobic respiration of the respiring tissue. So obviously we know that there's going to be a higher CO2 at respiring tissue, and that lowers the affinity of hemoglobin for oxygen. So more oxygen, but more O2 is dissociated. And that allows, this is just a bit of annotation, more aerobic respiration. So this is the Bohr shift. Now, if you're interested, the reason why hemoglobin loses its affinity for oxygen when there's more CO2 present is inside the red blood cell, the carbon dioxide combines with water to form carbonic acid, and that acid releases protons. However, that would make the red blood cell too acidic. So hemoglobin is a buffer for H+ ions, and when it binds the H+ ions from the acid, it dissociates the oxygen. So the hemoglobin is buffering the red blood cell from becoming too low at a pH, and a consequence of that is that the oxygen is released. Okay, so increasing, or there's more oxygen dissociation, or there's more unloading of oxygen when there's a higher carbon dioxide concentration, because hemoglobin has a decreased or lowered affinity for the oxygen, so it's being released. Now, higher carbon dioxide levels in the blood would mean there's a lower pH. So you have to link the idea of more carbon dioxide or a higher concentration with a lowered pH in the red blood cell, which causes the release of the oxygen for mark point number 1. Okay, so seals are diving mammals. They fill their lungs with air before they dive and hold their breath during the actual dive itself underneath the water. So we know mammals have trachea, bronchi, bronchioles, and then they have alveoli in the lungs, which are the air sacs, just like humans do. We're mammals as well. Okay, seals, although they can swim underwater for an awful long time, obviously they do not have gills, they're not fish, they do have lungs like we do. Right, figure 3 shows the dissociation curve for seal oxyhemoglobin and seal myoglobin. Okay, so hemoglobin is inside the red blood cells, and myoglobin is actually inside the muscle. Okay, so you could put here, myoglobin is a store of oxygen, and it has a very high affinity for oxygen. So the myoglobin binds oxygen and it remains bound, and it's a store of oxygen inside the muscle cell. And if you look at the sigmoidal shaped curve, okay, if you look at this bit here, this is myoglobin. The myoglobin only dissociates all its oxygen at very low ppO2. So at, for example, 4 kPa, the myoglobin will be pretty much 100% saturated. And it's only when that PPO2 gets very low that the myoglobin, the dotted line, will release its oxygen. So the oxyhemoglobin is the bold line, because obviously we do use the key. And at a PPO2 of 10 kilopascals, there's lots of oxygen associated with oxyhemoglobin inside the red blood cells. And the oxygen from the red blood cell, from the hemoglobin, will be released. So you can see here, very low saturation, because the oxygen is being released from the oxyhemoglobin. Okay, and that oxygen is then going to be used in aerobic respiration. So key point, take home message is any curve shifted to the left has a higher affinity for oxygen compared to adult hemoglobin. Any curve shifted to the right has a lower affinity for oxygen. For example, in the presence of more carbon dioxide. And it says, use the information in figure 3 to explain. Okay, so explain is what's the biology behind, to say why something happens, how the seal's myoglobin dissociation curve shows the seal is adapted for diving. Okay, so we've got to use that idea of the data on the graph, and we're going to say, why is the seal's myoglobin, okay, shifted to the left? What does that mean? And why is that, more importantly, an adaptation for diving? Okay, so this is the key bit of the question here. So when a seal dives, then obviously it can't breathe underwater, okay, because it closes its mouth. So there's no air going into its lungs, into the alveoli of the lungs anymore. It's like when you go diving yourself, you close your mouth, you go into the water. So any oxygen associated with hemoglobin, that's oxyhemoglobin, that will be dissociated at the respiring tissues where there's a low PPO2. So a low PPO2 at the respiring tissues, I'll just put respiring tissues, that causes more O2 to be released or dissociated. And that is used in aerobic respiration. Now, once the oxyhemoglobin has released all its oxygen, then the myoglobin, when the PPO2 is very low, then the myoglobin will release its oxygen. So it's a store of oxygen. Now, if there was no myoglobin, what would happen is the muscles of the seal would switch to anaerobic respiration in the absence of oxygen. Now, we know from anaerobic respiration, I'll just put this at the bottom, anaerobic, that does not involve oxygen, does not involve mitochondria. But what it does produce is increasing levels of lactic acid or lactate. So what happens with the oxymyoglobin up here is that because it's a store of oxygen, and that's then given off when the PPO2 is very low, it allows aerobic respiration to continue, which prevents the formation of lactic acid while the seal is diving for a short time anyway. So myoglobin has a higher affinity for oxygen compared to the seal hemoglobin, so versus the seal hemoglobin inside the red blood cells. The oxygen dissociates less readily because of the high affinity, or the higher affinity we should say. The oxygen associates more readily, was accepted. This allows aerobic respiration when diving at lower PPO2, or when the oxygen has been used up and fully unloaded from hemoglobin, and therefore it delays anaerobic respiration in the muscle cells of the seal, and it prevents lactate production. So take-home message, myoglobin is a protein. It's got a structure similar to hemoglobin. It binds oxygen with a very high affinity. The myoglobin is present inside the muscle cell, and you guys will do that a bit later this year, year 13. It's in the cytoplasm of the muscle cell. It's called the sarcoplasm. and it's only going to release that oxygen when there's very little oxygen available to that cell. So it's the last storage of oxygen. Okay, in question 9.3 then, we can do a bit of maths. So it says the scientists measured the oxygen carrying capacity of seal blood. They found the hemoglobin in the red blood cells in a 190 kilogram seal contained 1.07 times 10,000 centimetre cubed of oxygen. So we know this part of the question is going to be really important because it's saying 190 kilogram seal, and then it's saying that this seal used 5.2 centimetre cubed of oxygen per minute per kilogram of body mass. So that's 5.2 centimetre cubed of oxygen in 1 kilogram, and that is 1 minute. Well, we know that the seal is not 1 kilogram, it's 190 kilograms. So if you times 5.2 centimetre cubed by 190 kilograms, that will give us the volume of oxygen that the seal uses per minute, okay, obviously within that seal. So the 5.2 is per kilogram of body mass used up in 1 minute. So we times by 190 to get the volume of oxygen used up, not in, in 1 minute, but per seal. That's 988 centimetre cubed of oxygen used up per minute in this particular seal. Now, if the volume of oxygen was 1.07 times 10 to the power 4 centimetre cubed, and that is attached to hemoglobin, that's oxyhemoglobin, all that is available for aerobic respiration. So there's 988 centimetre cubed used up per minute in the seal. So you take the total volume of oxygen available divided by the volume used per minute, and that will give you the number of minutes the seal can dive by using the oxygen within its body. Now that comes out as 10.8 minutes. So a couple of scripts I saw, some students just put 10 minutes, but obviously you have to round up. You can either leave it as 10.8 or you can round up to 11 minutes. Or you could give it in 2 decimal places, that was okay. Okay, the next question then. Scientists investigated changes in the mass of fish from 3 populations of the same species. They used fish that had a life cycle of 1 year. Okay, so 1 year life cycle. Okay. Now the scientists set up 3 different fish tanks, A, B and C, each containing a separate population. So you've got to pay real close attention to this information. Each year the scientists removed all the fish from each tank and determined the mean mass of the fish, and they put back 10% of the fish in tank A. They put 10% the largest fish back. Tank B, they put fish back at random, and tank C, the 10% they put back were the smallest fish from the population. So the fish have a life cycle of 1 year. We're told that at the top. Every year they're going to take out all the fish, work out the mean mass, put back the top 10% of fish in A. So only the largest 10% are put back in A, and in C the smallest 10% are put back. Okay. Now in B, they don't take the top 10% largest or the bottom 10% largest. They just take 10% of fish without really looking at if they're the biggest or the smallest. So that's kind of random. So we know tank B is going to act as a control to look at the effect of putting back the largest 10% of fish, or looking at or comparing with the smallest 10% of the fish. So you always use a control in any experiment, and this will always give you a baseline value against which you can then compare. So tank B is our baseline control value against which we can then compare tank A with tank C and the mean mass of those fish. So if we look at the data then, okay, 1 year life cycle. So they put the fish into the tank A, B and C at the start. And hopefully you can see that the starting mean mass is actually very similar for tank A, B and C. So at time point 0, the masses are between 3 and 3.5 grams. Sorry, the mean masses. Now after 1 year, okay, we can use the key. So we've got tank A. So the mean mass is just over 4. So it's 4.1 grams. They put back the top 10% of the fish in terms of their size, the largest fish. And it's kind of very similar. The next year, it's just under 4 grams. They take the top 10% of the fish, the largest fish, put them back. And this time it's 4.7, 4.8, okay? And then it's going to remain constant. So what we can see with the large fish when they're put back is that there's an increase in the mean mass over subsequent generations in tank A. Now, if you look at tank C, when the 10% smallest fish are put back, we can see over subsequent generations that the mean mass goes down. Now, each tank is separate. So tank A, we're only really looking at the largest fish being able to breed. Tank B is the control, and tank C, only the smallest fish are being selected for. So it's artificial selection because obviously we're controlling which fish are being put back into the tank, which can then breed together. But because we're selecting for one extreme, it's actually directional selection. Because in tank A, you're selecting for the largest fish and they're allowed to breed and their mean mass increases over many generations. In tank C, you're selecting for the small extreme and you're allowing the smallest fish to breed together, which actually we see decreases the mean mass of fish over a number of generations. Okay. So what type of selection were the scientists modelling in the investigation by putting back only the largest or only the smallest fish in tank A and C? So it does say or, so I'd highlight that. Okay, tank B, large and small fish might have been put back together as part of the 10%. So it has to be directional selection, okay? Because one extreme, not two extremes, but one extreme is being selected for in that particular tank. So in tank A, we said the large extreme is being selected for, and in tank C, the small size extreme is being selected for. So one extreme only is allowed to breed in that tank. Okay. Now, just for you guys then, this is directional selection. The other two types of selection that you must learn are disruptive selection. Okay, and that's where two extremes are selected for, okay, within the same population. And then you've got stabilizing selection, where the mean or the average phenotype is actually selected for. So the existing mean phenotype is selected for. So the population number with the mean phenotype or close to the mean phenotype would increase in population, and the two extremes in stabilizing selection would be selected against. So actually the standard deviation around the mean would actually increase here. Sorry, the standard deviation would decrease, sorry. Put that right way. Okay, explain the purpose of tank B then. So we know tank B is a control, and the control provides us with a baseline. That's how science works. So how science works. Now, anytime you get a control and a baseline value, that's needed, because then you can test the effect of whatever factor you're investigating. So in this case, we can test the top 10% of fish breeding together in terms of the large fish, or we can compare the 10% of the smallest fish. So to show the effect, or to compare with tank A, tank C. So there'd be no selection in tank B. That's our control. Okay, calculate the ratio of the mean mass of fish removed from tank A to the mean mass of fish removed from tank C at 1 year and at 4 years. So if we look at the graph, okay, it says the ratio. Now you need two numbers for a ratio. Mean mass, so we've got mean mass on the y-axis from tank A. So we've got tank A here, and to the mean mass of fish removed from tank C. So tank C is the dotted line there, at 1 year and at 4 years. So each ratio we're going to have two values. So we'll do 1 year first. So we'll go 1 year, and it's saying tank A 2, so it's A 2C. So to get this ratio, we go A divided by C to 1. So if A was 12 and C was 6, 12 divided by 6 is 2 to 1. So the ratio is 2. So if we go to 1 year, we're going to go that value and that value. So that is 4.1 at the top for A and 3.4 to 1. So you guys can work that one out. The second ratio is at 4 years. So again, we're going to circle A at the top, and that is 4.8. And tank C is 2.4 to 1. So that equals 2 to 1. Okay, so you've got your two ratios. Then the first value, okay, we can see how many times bigger is that than the second value. Okay, so you just do a division. Okay, so what you should have had is 1.2 to 1, and the second value is 2 to 1. And then if you divided the two numbers together, you would have found that it's 1.6 times bigger, and they accepted 1.7 if you rounded up. So 1.6 times bigger. So if you go 2 divided by 1.2, give you 1.6. Okay, sea fishing is regulated in law. The size of the mesh used in some fishing nets is controlled so that small fish can escape but large fish are captured. This regulation is designed to protect populations of wild fish. Okay, so using all the information, so you've got to go back to all the information, use that. Evaluate, this is AO3, where the scientific investigation supports the use of these types of nets in sea fishing. Okay, so let's just have a look at the information then. It says this regulation is designed to protect populations of wild fish. So populations of wild fish. Okay. The size of the mesh used in some fishing nets is controlled so that the small fish can escape. So the small fish escape, and they can, you can write this down, breed. So they're allowed to escape, so they can still go off and they can breed. The large fish obviously being captured within the net, they can't get through the little holes in the net, so they're going to be removed, and obviously the farmer's going to, the fisherman's going to collect them and sell them for profit. So what do we know about breeding of small fish over time? Well, from our experiment in the tank, we know that the small fish, when they breed together over generations, that's going to decrease the mass of the fish over time. So that was the directional selection that we said earlier. Now, if you think about that, we've shown through the investigation that the mean mass is going to decrease over time if the small fish are allowed to breed together, directional selection. What we're not told is the population of the fish in the tanks. It just gives us the mean mass of the fish over generations. So some of the missing data here is that we've got no data for populations of fish in the tanks. We've just got the data for the mean mass. So population numbers are not given in data, only mass. So we know how the small fish, when they breed together, affects their mass, but we're not given any data about the population number or the yield of these fish. So that's an important thing to remember. So let's have a look at some of the answers then. The other thing you would have noticed from the graph, there's no standard deviation bars, there's no statistical test done. So we actually don't know if the increase or decrease in mass is significant or not. Okay, so sea fishing reduces the mean mass of fish. So that's what they're saying there, because the small fish are allowed to escape through the small holes in the net. And when small fish breed together, we know the mean mass decreases over time. So that is acting like tank C. So tank C was mimicking the effect of the small fish being able to escape and breed out in the wild. However, the information that we're given in the experiment is only for a particular species of fish. Okay, so we know this particular species of fish, when it breeds together and it's small, the mean mass goes down. We do not know, there's no data, that's missing data, if that is the case for all different populations of wild fish. So in AO3, one of the techniques is to look at this experiment and what species is the investigation actually done on. So you've got to pay careful attention to that. And what does it say in the conclusion in relation to the different species? Is it saying all species in the conclusion? But maybe the data is only for one specific species. Okay, so only one species used. Sea fishing catches other different types of fish. So it's saying in the nets, okay, maybe it's not one species, but maybe they catch a collection of different species of fish in the nets, and the little ones from each of the different species might be able to escape. Well, we've got no data on the other species. No stats test, no p-values. We don't know if the differences in means are significant or not. The size of the tank may affect fish growth. So we actually don't know the size of the tanks that were used. We're going to assume they're all the same size, but that could also affect the growth of the fish. Now, the other thing that was mentioned in the question was that the life cycle of these fish is 1 year. That was given at the very start of the question that we circled. Now, we don't know the life cycle of the wild fish, so it could be a longer life cycle. Therefore, that doesn't match the data that we have, because we've only got the data for fish with a year-long life cycle, where we've taken the biggest or the smallest after breeding through that year. Okay. So that was the last one down here. Look, sea fish do not have a life cycle of 1 year, or we do not know the reproductive life cycle of the wild fish necessarily. And also the sea fish might not reproduce all at the same time as they would do within the tank when they're all put back together. Now, point number 7 is quite interesting. It says there's no measure of the number of fish, so there's no population number. So no population number, only the mean mass was measured. Okay, so question 11.1 then, it says, describe the biochemical test you would use to confirm the presence of lipid, non-reducing sugar and amylase. So a little bit of annotation first. So lipids might be triglycerides, and we know that's the emulsion test. Emulsion test there. Now we know non-reducing sugars, the example you guys have to know is sucrose. So we need to do something to sucrose first because it's a non-reducing sugar. And then the last one is amylase. Well, we know amylase is an enzyme and therefore a protein. So if we look at the lipid emulsion test, if you were given a food type like seeds, you'd have to crush them up first and then you add ethanol or a type of alcohol. Can you say you shake it and you mix it? Then you add water, cold water, and you shake it and you mix it. So it's very important you get this correct with the detail. So crush the sample of food, add ethanol, shake it and mix it, then add water and shake it or mix it. And after you've added water and give it a shake, you might see a white emulsion. Okay, so a white emulsion is the best thing to say, white emulsion. If you put cloudy emulsion, they didn't like it. And if you put precipitate, obviously they didn't accept that. Right, there's no heating in the emulsion test. There's definitely no precipitates formed. Now for a non-reducing sugar then, obviously sucrose is going to give a negative test if you just added Benedict's reagent and heated to 95 degrees C. So there's 1 mark for saying if the Benedict's reagent remained blue, that'd be a negative result. So what you have to do is you have to boil the sucrose with dilute acid. Then you have to neutralize with a weak alkali like sodium hydrogen carbonate. Once the pH is back to pH 7, you then add Benedict's reagents and you heat to 95 degrees C. Now because you've got an equal concentration of fructose and glucose, they're two reducing sugars, they're monosaccharides, we're going to get an orange or brick red precipitate. So the heating is to 95 degrees C. Now just bear in mind, with different reducing sugars, depending on the concentration present, you can get different coloured precipitates. So a low concentration is going to be a yellow precipitate, then green, then orange, and then brick red precipitate. So sucrose is a non-reducing sugar. It'll give you a negative result if you just heat it to 95 degrees C with Benedict's. It'll stay blue. Boil with dilute acid, that breaks the glycosidic bond between fructose and glucose, separates the monosaccharides. They are now reducing sugars and you'll get a positive result, precipitate. Okay, just bear in mind for the sucrose then, just put this at the bottom. So sucrose is made from alpha glucose and fructose. If you had 1 molar, this is really important, 1 molar sucrose, and you added a dilute acid and hydrolyzed the bond or broke the bond, the glycosidic bond between the two monosaccharides, you would have 1 molar glucose and 1 molar fructose. So you would have double the concentration of reducing sugar compared to the concentration of sucrose. So that's really important. It's just like saying if you had 1 molar maltose and maltase was added to hydrolyze the bond, you'd have double the concentration of reducing sugar because you'd have double the concentration of monosaccharide. Okay, then the protein is amylase, it's an enzyme. You add Biuret, you do not heat, and you'll get a blue colour to a purple colour. So no heating and no precipitate, it's just a pure colour change in the liquid. Okay, last question then. Describe the chemical reactions involved in the conversion of polymers to monomers and also the opposite, monomers to polymers. Okay, so we know monomers, really important to describe this correctly, are very similar or identical repeating molecules that are linked via condensation reactions to form a bigger polymer molecule. Now, during the condensation reaction, the bonds are being made, so you have to reference those. Now, in hydrolysis, the polymer, the larger polymer molecule, the bonds are being broken by the chemical addition of water, and that separates the monomers, which we know are identical or very similar, okay, into the individual monomers. Then it says give two named examples. So we're going to give two specific examples from the biological molecules booklet that you did at the start of year 12. So look at the answers then. So condensation reaction joins monomers together into polymers. It forms chemical bond and releases H2O. Hydrolysis is the opposite. The polymer is broken down, so chemical bonds are being broken here between the monomers to release the monomers, and it uses up water. Examples then of polymers include starch, which is made from thousands of alpha glucose, and starch has glycosidic bonds, but be careful, it has alpha 1 to 4 and alpha 1 to 6. Okay, so starch is made from amylose and amylopectin. Amylose is a coiled polysaccharide, and the amylopectin has branches coming off it like that. So the 1 to 6 glycosidic bond is the branch point on the amylopectin of starch. You could have had cellulose. So we know cellulose is made from thousands of beta glucose. Now each adjacent beta glucose is rotated or inverted 180 degrees. Please bear in mind that cellulose is not coiled, it's not branched, it is made of a straight chain. So the straight chain is one molecule of cellulose. Now we know the straight chains of cellulose hydrogen bond and cross-link together to form microfibrils that gives the plant cell wall its strength, a high tensile strength. You could have had the polynucleotides in DNA and RNA. So DNA is double-stranded. It's got two polynucleotide strands where nucleotides are linked together in a 5' to 3' direction. RNA has RNA nucleotides with ribose, uracil, adenine, guanine, and cytosine as the nitrogenous base. And the RNA nucleotides are linked via phosphodiester bonds into that single strand, again in a 5' to 3' direction. So any suitable example would have got you full marks on that one. Okay, the point actually that you do need to know is a dimer, like a disaccharide or a dipeptide, is not a polymer. So technically a polymer is 3 or more monomers linked together by covalent bonds. Most polymers are made from thousands of monomers linked together, like starch, cellulose, glycogen. Proteins are also polymers. They're made from amino acids via peptide bonds. Proteins can range from anywhere between 10 amino acids up to hundreds, if not thousands, of amino acids. So proteins can be different lengths.